for BALL. So I'll just, I would like to mention that this is a translational project, which is bench to bedside project, where the laboratory outcome is directly applicable to the clinical site, and this is not a basic basic science research. So I'll just briefly introduce what is CAR and what is CAR T-cell therapy. So CAR stands for chimeric antigen receptor. It is artificial receptor which is artificially expressed on the T lymphocytes of the patient or of the individual. So this CAR T-cell therapy is a combination of cell and gene therapy. So it is specific to a particular antigen which is present on the cancer cell. So this CAR T cell is targeted to a particular cancer antigen which is present on the cancer cells. And here is the SCF. If you see, this is the CAR T cell, and here the receptor is CAR receptor, which has different domains. For example, the SCFB domain, which has an antigen specificity, and the hinge domain, transmembrane domain, and the endo domain, which has CD3 zeta and the 4-1BB co-stimulatory signal. So the entire chimera of the protein is CAR, and this is the autologous treatment. Autologous treatment where where the patient, if we see this the right side image, so where the CAR T cell therapy has multiple steps, and some steps are done in the clinic and some steps are done in the laboratory. So for example, the first step is the apheresis of the patient. Like we take out the mononuclear cells from the patient and bring that mononuclear cells into the laboratory. There we isolate the T lymphocytes from the blood and then activate those T lymphocytes. And after activation, we modify these activated T lymphocyte with the help of lentiviral vector. It is a kind of gene delivery vehicle. And now, after this modification, this process is called transduction. So after transduction, our CAR T cells are ready, and after that, we expand these CAR T cells in enough number, and then again we send these CAR T cells to the clinic, and then reinfusion occurs there in the clinic with the help of some chemotherapeutic drugs, for example, fludarabine and cyclophosphamide. These are some lymphodepleting drugs which deplete the lymphocytes and give enough space to expansion of these CAR T cell in the patient, and then there in the body, these CAR T cell will kill the tumor cells and the remission will occur. So this is the entire CAR T cell therapy picture. So now, so I'll just briefly introduce what is this CAR receptor. Has different domains. It has different functions. For example, the SCFB domain, single chain variable domain. So this SCFB domain provide the specificity. This SCFB domain provide the specificity to the antigen, targeted antigen. Also, we can tune this SCFB domain against antigen to increase the efficacy of the CAR. As well as this SCFB domain determines the immunogenicity of the CAR. For example, if this SCFB domain is derived from the antibody of the murine origin, so it can cause immunogenicity. But if it is derived from the antibody which is originated from the human origin, then it will not cause immunogenicity. So this SCFB also determines the immunogenicity of this CAR. And the second domain is the hinge domain, which provides the flexibility and length to the CAR, and also it helps in the positioning of the CAR on the surface. And this transmembrane domain, it provides the stability, as well as it also helps in release in the cytokines after the activation and binding to the targeted antigen. And then next domain is the intracellular domains. So the intracellular domains, there are two domains, basically the CD3 zeta and the 4-1BB, which is a co-stimulatory signal. So both signals provide the antigen activation signal for the proliferation of these T cells. And apart from these different domain function, there are some cytokines which is secreted in the environment. They also regulate the differentiation of these CAR T cells and also help in the expansion of these CAR T cells. So with this overall this regulatory determinants of the CAR function, so we published recently in 2009 in the Frontiers in Immunology. This is a review article which we published in our lab, from our lab. And So now coming to the CD19. So CD19 is the antigen which is present on the cancer cells. It is a promising blood cancer target. Like most of the, like if we see here, the data says that like 70% clinical trials as well as the basic research of the CAR-T are targeting CD19 antigen. So why like other targets are also like in focus, but the CD19 is like more focused. So why CD19 is a promising target, I will explain in the next slide. So here, if we see the B cell development, and it's associated malignancies. So this is the development stage of the plasma cells, like B cell development stage, from bone marrow to the periphery and then again in bone marrow. So if we see this CD19 surface expression, so this CD19 occurs from pro B cell to the late plasmablast. So in between, if any of the cell become cancerous or tumorogenic, then all these cancer or leukemia or lymphoma, they can be targeted with CD19 targeted antigen, CD19 targeted CAR T cell therapy. So this is why this CD19 is a promising target, and people are like multiple clinical trials are ongoing with this CD19 antigen. So now coming to the clinical trials which is happening for CD19. So here I have shown only 3 primary institutes which are in collaboration with different pharmaceutical companies. They are like they have designed, and they are also they have run multiple clinical trial, phase 1, phase 2, and they all 3 they have targeted CD19 antigen, and different patients. Like if we see, like here, approximately 100 patients they have taken and for this trial, and they have seen very promising result, like around 90% complete remission they have seen with this therapy. So like if we see this UPenn group, they have in collaboration with Novartis, and this NCI, they have collaboration with Kite Pharma, and this MSKCC, they have collaboration with Juno Therapeutics. Like multiple academic institutes, they are in collaboration with different pharma companies, and now bringing these CAR T cell to the market. So looking at this, these are the different five CAR T therapy, approved CAR T therapy, which is available in the market. The first one was approved in 2017, was the Kymriah, which was from Novartis and developed by the UPenn group, and it is approved for relapsed and refractory BALL. They also target CD19. Now it is approved in different countries also, like apart from USA, Europe, Japan, Canada, and Australia, and and the cost of this therapy is approximately $500,000. Now coming to the second therapy, second drug, which is Yescarta, developed by the Gilead and developed by NCI group. So this is approved for the relapsed and refractory DLBCL. This also targets the CD19 antigen, which is also developed after a few months after this Kymriah is approved. It is also approved, and recently in 2020 and 21, these 2 CD19, Tecartus and Breyanzi, these 2 are also approved, and the cost is around $500,000. And very recently in March 2021, so another CAR T therapy which targets the B cell maturation antigen, which is for multiple myeloma. This is a cancer of the plasma cells. So this is also approved. So these are the multiple products which is approved in the market. So now we see the current challenge of this CAR T cell therapy in Indian perspective, like blood cancer. So around 10,000 to 25,000 new patients of pediatric cancer comes every year, and more than 7,500 new cases of childhood cancer are registered every year, and 6,000 cases will be like BALL in that. And if we see from the conventional therapy, around 70 to 80% complete remission is there, but the problem is the relapse. Around 20 to 40% patient relapse with the existing therapy, and and in that relapse, only less than 10% patients undergo allogenic transplantation. So like other patient, they, like, if we can say that majority of the Indian patient choose palliative care, and they ultimately die because of the unavailability of this therapy. And also, if we see the challenges of the CAR-T, which is the major problem, is the affordability. Like the current cost of this CAR-T is around $500,000, and we can say it will be unaffordable to majority of our Indian patient. And the lack of infrastructure as well as the technical skill. So currently, the available approved CAR-T therapies, if we see the manufacturing of these drugs, around 50,000 to 75,000 per patient, the manufacturing cost is there, and in that, around 60% total cost comes from the skilled manpower. So this therapy needs proper skilled and technically skilled manpower to develop this. So these are the current challenges of CAR-T therapy in Indian perspective. So looking at these challenges, my major and like aim objective was to develop the indigenous CAR-T platform. CAR-T on these are my objective, like first, design and development of CD19 CD28 CAR to set up the entire platform. After that, design of the humanized CAR and its validation in by computational modeling, as well as validation in the relapsed and refractory BALL patient. And then after that, preclinical like efficacy, toxicity, and tumorigenicity. And after this preclinical validation, the next objective was to develop a patient scale CAR-T cell manufacturing to reach to the clinic for first in clinical trial as per the industry standard. So I will go from each objective one by one. So like the first objective was to design and develop an entire CD19 CD28 like develop this CAR, anti-CD19 CD28 CAR with healthy donor, as well as its validation with relapsed and refractory BLL patient. So why I have designed the CD19 CD28 CAR? Because at the first time I need a product.
Only for me.
Yes, sir.
I think your sound went and the screen hung.
Okay, so now it is coming.
Yeah. Please go ahead.
You're fine.
Oh, yeah. So here I'll go. So first objective was to design and development of anti-CD19 CD28 CAR with healthy donor and its validation with relapsed and refractory BLL patient. So if we see the CAR-T generation process, there are multiple steps, like 4 steps. First is the design of the CAR construct. And after that, production of the gene delivery vehicle, which carries the CAR construct. And after that, manufacturing of the CAR-T with the transduction of this lentiviruses, and then functional characterization of this produced CAR-T. So in that, so firstly, we have designed a CD19 CD28 CAR, which was derived from the NCI, and we did some codon optimization to increase the surface expression, and also we used different promoter, highly like EF and alpha promoter. It is a strong promoter which increased the surface expression of this protein. And if we see the amino acid similarity of this CD19 CD28 CAR is 100%, but nucleotide similarity is 0%. And so the major aim to design this CD19 CD28 CAR, which was derived from the NCI, to develop entire protocol from the scratch, designing to the functional validation. So we have taken this this CAR construct from the NCI and did some modification. After that, designing. So we optimize different lentiviral production, like multiple steps which is involved in the lentiviral production. So the first step was, so whether we should use second generation or third generation lentiviral system. Here we have used different like both system. Also the packaging cell like LentiX293 T cells as well as the HEK293 FT cells. All these packaging cells also we have used to get the high titer. And apart from that, next we also like we also isolated and quantified the plasmids, as well as the like how much plasmid ratio should be there to get the high titer, and which media should be suitable for getting the high titer. And after this plasmid and media optimization, like how much incubation time should be there, like for the virus, like in the virus production we do co-transfection of the plasmids, like multiple plasmids. So how much co-transfection should be there, like how much incubation should be there, different time points we have optimized, like 6 hours, 16 hours, and 24 hours. And after that, like how much collection time, which collection time point should be suitable to get the high titer. So that also, and the concentration method, like we have used different methods like poly-lysine, ultracentrifugation, and different speeds also to get the concentration of the viruses. And after the concentration, there should be purification of these viruses, because these unpurified virus will indirectly will kill the T cells. So we should also purify these viruses. So here, so we use different method like sucrose, cushion, as well as 100 kDa anicon filter to purify these viruses. And after this, all this optimization, we were able to achieve this around, like we can say on average 200 million TU per ml. We were able to achieve the lentiviral titer. And after this lentiviral optimization, we optimized the CAR T cell generation with this lentiviral vector. So first step was the which method we should use, spinoculation or retronectin mediated transduction, and which method will give higher transduction efficiency. So here we have optimized these two methods, and finally we chooses a spinoculation. And different volume of the reaction, like in which plate we should use, like different reaction mixture also we have optimized. And then which cell density and what is the MOI to get the good transduction efficiency, and what is the bead and cell ratio, like which ratio should be suitable for the activation, and the IL-2 concentration and the time of activation. All these steps, multiple steps we have optimized. And purity of the T cells, like if we activate the T cell, only the PBMCs, then how much transduction efficiency will be there. If we sort these purify the T cells, how much transduction efficiency will be there. So all those steps I have optimized. And after that, like which purification method is suitable for getting the high transduction efficiency, that also we have optimized. So after this optimization, all these multiple steps optimization, the transduction efficiency we got from the range 2 to 35%. These are the, like during the optimization we got this transduction efficiency. And after that, so this is the proliferation of these CAR T cells. And efficacy, here we got around 100% killing of the CD19 positive target cells. For example, NAM6 and Raji cells both are CD19 positive tumor cells, representing the tumor cells, CD19 positive cancer cells. And K562 is a CD19 negative cancer cell line. So here in K562 we did not see any BLL relapse, and who has not undergone any stem cell transplantation were recruited also.